This is the first in a series of screencasts on the neurophysiology content of PM1A. So the lecture aims of this series of screencasts are to tell you a little bit about neurophysiology, and also, as you'll see here, to link it with content going forward into other therapeutic areas, which you'll deal with later on in the course. So what we're going to do in this set of screencasts is to hopefully to tell you a little bit about the basic divisions and the organization of the nervous system, and particularly the role of the central nervous system. To understand that neurons are the functional units of the nervous system, to understand the electrochemical basis of what we call the nerve action potential. So we'll see that a number of ions are important, in particular things like sodium and potassium ions, and also to understand action potential propagation and the role of this insulating material called myelin. This will cover probably the first 3 to 4 screencasts, and then I'll come back with some more lecture aims after this. So we basically break this neurophysiology content down into probably 5 main sections, the first of which is organization of the nervous system. Essentially, the nervous system has 2 major divisions. So the central nervous system, consisting of the brain and the spinal cord. The spinal cord is a very important part of the central nervous system. We would appreciate probably that the brain is part of the CNS, but also the spinal cord. Everything outside of the central nervous system is called the peripheral nervous system, or the PNS. And this can be divided into 2 main sections, the automatic or autonomic nervous system, and something called the somatic or the motor nervous system. And we'll deal with the autonomic nervous system, the ANS, and the somatic nervous system in the final few screencasts in this series. So let's start off with the central nervous system. This is a very basic diagram, probably of a lower invertebrate perhaps, that shows that the central nervous system is split into 3 main basic regions. So the forebrain, the midbrain, and the hindbrain. And these have a little bit more scientific names, if you like. So the prosencephalon is the forebrain, the midbrain, the mesencephalon, and the hindbrain, the rhombencephalon. And these contain important regions for neurobiological function that we'll find about as we go through this set of screencasts. So the forebrain is the main part of the brain, if you like, contains the large brain or the cerebrum and a sense processing organ called the thalamus. The midbrain, the mesencephalon, contains the cerebellar peduncle, which are really just the areas where some of the nerve tracks ascending and descending from the forebrain to other parts of the hindbrain and down through the spinal cord into the peripheral nervous system are located, and areas such as the substantia nigra. The substantia nigra we've highlighted here because it's quite an interesting area. From a medical point of view, the substantia nigra is associated with Parkinson's disease. So one of the characteristics of Parkinson's disease, if you get degeneration of this area here called the substantia nigra, and some of these nerve tracts look black, hence the name nigra. And if they degenerate, that's often a sign of things like Parkinson's disease. And we'll see that there are some imaging techniques that we can use that can maybe alert us as medical professionals of actual or impending diseases. So in particular, as I said, Parkinson's disease, other areas of the brain, perhaps Alzheimer's disease. We'll see some examples as we go through. The hindbrain, as I said, maybe contains some of the other regions, in particular the pons, which is a bridge between maybe the hindbrain and the midbrain, the medulla, which contains lots of nuclei that are important in things like breathing, and also the little brain or the cerebellum, which is intimately involved with movement. So this is, if you like, perhaps an old school diagram of a brain taken from one of the original textbooks, Gray's Anatomy, I think, which shows you some of these areas how they've developed in humans. So this area here will be the forebrain, the midbrain is in here, and the hindbrain down here. So you can pick out some of these areas referred to above and in the slides over the next few slides. So whereas this is quite maybe an old school diagram, here we show maybe a modern version of this diagram, showing again the cerebrum. In here is our midbrain, and down here is our brainstem, containing these important areas that I alluded to before. Just to highlight a few of them, the thalamus, shown in here, is what we call our sense processing center. So a sensory function, sensory information from the peripheral nervous system, which enters through the spinal cord, processed in the thalamus, and then that information is communicated mainly with the cerebrum, and it tells us how to maybe react to different sensory inputs in the central nervous system. The hypothalamus, shown down here, is another important region, regulates endocrine, the endocrine system, which you'll hear about in other lectures maybe, by releasing hormones or communicated via hormones released from the pituitary gland, again shown down here as an extension of the hypothalamus. So we could have picked a lot of things maybe to highlight in this diagram. These are some of the important ones. As I said, we'll hear about these a little bit more as we go through the screencasts. So yeah, maybe as I said, I've alluded to the fact that if as medical professionals we want to have an early diagnosis of disease, particularly brain diseases, obviously in this case, then we can use different techniques. For brain disease, one of the most common set of techniques we use are imaging techniques. So a number of techniques. The first one shown here is PET scanning, which you may have heard about. PET stands for positron emission tomography. And essentially it is a scan based on radionuclide elements which emit rays, in this case gamma rays, which can be scanned and give us information about the brain. So hopefully this is what your brain looks like, a nice red active normal brain shown in a PET scan. So these areas of red show high activity, maybe the blue areas are showing lower activity. So in a PET scan, hopefully if you have a PET scan, this is what your brain looks like, nice and active. These areas probably correlate best with blood flow. However, if we have a diseased brain, in this case, this is a person that unfortunately has Alzheimer's disease, we can see that the PET scan is very different to normal. So instead of lots of these nice red active areas, we have these blue cold areas which indicate some kind of degeneration of the neurons and the nerve tracts in these areas. So if somebody has got probably late onset Alzheimer's disease, they'll look like that. However, mild to moderate Alzheimer's disease, maybe they look somewhere in between this nice normal scan and this degenerated brain. And if we have a scan, maybe we as medical professionals we can intervene with therapeutics ahead of the patient reaching this situation. So scanning is important. PET scans and also other scans like fMRI or MRI. And MRI stands for magnetic resonance imaging. If the F is in front of it, functional magnetic resonance imaging. Again, this measures changes in blood flow relative to neural activity. So a normal brain, this is shown in grayscale typically now. This is what a normal brain looks like. Lots of areas of white and grey. But in a diseased brain, again, an Alzheimer's brain, you can see lots of degeneration. So instead of this nice full white and grey areas, we get these areas of degeneration, probably to what we call cholinergic nerve tracts. So nerve tracts that carry the neurotransmitter acetylcholine tend to degenerate in Alzheimer's disease. And again, if you can scan somebody who's got early onset Alzheimer's disease and they hopefully haven't progressed to this stage, but don't look as nice as a normal brain scan, fMRI brain scan, then maybe you can predict disease and maybe you can intervene. So yeah, you've heard about these terms. Maybe we'll deal with them as we go on through the screencast. White matter and grey matter. So using the old grey matter is something we may have heard about. There is some scientific relevance to that. Grey matter are our nerve cell bodies, and the white matter are nerve fibres. And we'll see later on that this is because of an insulating substance called myelin, which tends to speed up nerve conductions. This is just a couple of slides now taken from Columbia University, just showing some of these areas in a little bit more detail. So highlighted in red here are areas of the midbrain. And again, we can look at models like this to make sure that everything is functioning and looks normal. Here on another fMRI scan, this is the area corresponding to the midbrain. So again, if we look at this area, check it for abnormalities. If we've got access to neuroimaging, we can say something about the any potential diseases to the brain. So scanning, imaging is very useful for medical professionals. Similarly, this is the hindbrain, so we've highlighted some areas. This in red here, this is the pons, the bridge between the cerebrum, the big brain, and the cerebellum, the little brain. This is something called the medulla oblongata, and this is an oblong or rectangular region that's very important for some of the nuclei that are involved in controlling physiological functions, which you'll hear about in other parts of the course. So things like the respiratory nuclei, and nuclei that control blood pressure. And the cerebellum is shown here, very important for things like movement and balance, diseases such as ataxia, where you lose coordination, are due to deficits in this area. So as I said before, if we have a scan and we show that there's degeneration in any of these areas, then maybe we can predict disease. Again, maybe a textbook image taken or taking a MRI scan and making it into a nice pictorial image. We can say something about the anatomy and physiology of the brain. In particular, things to point out here, the brain is surrounded by tough membranes or meninges, three main types of meninge. Sorry, let me just go back to that. The dura mater, the arachnoid mater, and the pia mater. Those are shown here. So you can see this is the arachnoid mater, this is the pia mater in here, and this is the dura mater. So again, you can have a look at this in a little bit more detail after the screencast. Essentially different membranes that surround the brain, but also help the separation of the different parts of the brain from this blue cerebral spinal fluid. So cerebral spinal fluid, CSF, lies between the arachnoid and the pia mater, very important in cushioning the brain, so preventing the brain from physically banging against the skull bone, but also very important in providing nutrients and oxygen to the brain. So it has a hemostatic function. The spinal cord, again, if we look in a little bit more detail, if we were to take a transverse section through the spinal cord, we would see, or the spinal column, we'll see that it's got four of these main horns. The upper horns in this case are called the dorsal horns, and the lower two horns, again in this case depending on the orientation, are called the ventral horns. What's probably important here is that this area is where the delicate spinal cord passes. Obviously it has to be protected through bone and these horns, and maybe importantly for neurophysiology, these horns are the areas that either take nerve fibers into the central nervous system or out of the central nervous system, and hopefully we'll see that on on the next column in a next slide in a little bit more detail. So essentially, in these four horns, the dorsal and the ventral horns, there are 4 spinal nerves per segment. These are involved in communication from the peripheral nervous system and also in reflexes. So probably importantly, take-home message is that the dorsal horns, the dorsal roots, are important for what we call afferent information. So afferent just means information that's going into a system. So information from the peripheral nervous system, things like pain and the sensory information, enter the central nervous system via these dorsal roots, which feed into the dorsal horn. Conversely, efferent fibers are ones that come out of the central nervous system to the periphery. So efferent fibers exit, this time via the ventral horn and via these ventral roots. And this is important in information to things like the somatic nervous system and the autonomic nervous system, which we'll cover in some of the later screencasts. So that's a good place to end the first screencast. What we'll do after this introduction is go on to talk a little bit about neurons and also how neurons communicate within the central nervous system.